Thank you for inviting me to give this presentation, and I think we should set off right away. So the overview of my talk is that I'm going to ask the question of what genomics has told us about schizophrenia, about the nature of the disorder. So I'm going to review what we already know about schizophrenia, which there's quite a lot, and I think this kind of gives us some prior expectations for genomic studies. I'm going to overview genomic findings in schizophrenia in the context of what we know from other types of research about schizophrenia, and I'm going to try and ask the question: what do genomic studies tell us about schizophrenia? Particularly, how does it happen? When does it happen, and where in the brain does it happen? And finally, I'm going to talk a bit about what I think we need to do next if we're going to have decent impacts on clinical practice. So schizophrenia is a common psychiatric syndrome. It's a collection of signs and symptoms that clinicians over more than 100 years have noted come together and predict certain outcomes. It's not a disease defined by a pathophysiology or anything else other than its clinical signs and symptoms. There are basically two classes of symptoms: positive psychotic symptoms. These are usually episodic. They typically have their first onset in adolescence or early adulthood. They consist largely of delusions, which are fixed false beliefs, which can be bizarre in schizophrenia, hallucinations, which are perceptions without stimuli, often auditory hallucinations, hallucinatory voices, and disordered thinking. And these positive symptoms occur, as I say, largely episodically against a background of negative symptoms, which are typically chronic and indeed can predate the onset of psychosis. And these include things such as impaired motivation, reduced spontaneous speech, and social withdrawal. And it is these negative symptoms that are particularly socially disabling. We also know that there are fuzzy borders. Schizophrenia, there is no clear distinction between schizophrenia and other psychiatric disorders, or in fact, wellness. There's particularly fuzzy border with bipolar disorder, so much so that there's this entity called schizoaffective disorder that is a kind of admixture of the two that sits between them. Another feature is cognitive impairment. This can predate the onset of psychosis or occur after and potentially progress. There's great controversy about whether it progresses, but the important things is that cognitive impairment, whenever you start to see it, affects multiple domains. This shows different domains of cognitive function, and it affects most domains tested. There's not a sort of nice signal in one particular cognitive domain. And in fact, this study from one of our students compares schizophrenia with bipolar disorder and schizoaffective disorder. And what you see is that there's this gradient in that schizophrenia is most strongly affected, bipolar disorder less strongly affected, although there's still impairment, and schizoaffective disorder in the middle. There's also reduced fecundity, and this just shows the fertility ratio for male and female patients with schizophrenia, and this is an important thing to take into account when thinking about the genetics. It has a big impact on the individual. A large number of people with a diagnosis of schizophrenia are unemployed. Life expectancy is reduced by 10 to 20 years, and it costs a huge amount, over £12 billion per year in England. It can come on suddenly, but more usually comes on slowly, and when it can, and sometimes quite insidiously in late adolescence, early adulthood. It doesn't always have its onset then, but that's typically when it occurs. But it has a highly variable course and outcome. In fact, there's a paucity of good, robust longitudinal data. But what we, and this is, I would take these numbers with a pinch of salt, but this just describes different potential life courses of someone with schizophrenia. Someone who has one episode and then recovers, repeated episodes with recovery between, repeated episodes with impairment, chronic impairment, and repeated episodes with worsening impairment. But the point is, when you see someone at the beginning with schizophrenia, it's very difficult to tell whether they're going to be walking out of that admission and they'll never be ill again, or whether they're going to have a lifetime of quite severe psychiatric problems. The other important thing for this talk is that because we sample patients typically for large genomic studies from clinical samples. We tend to sample prevalent cases that we're seeing in the clinic at the time, which are highly enriched for cases with poor outcomes. So a lot of the genomics has been done on people with the worst sort of outcome, 50% of the people with schizophrenia. The pathophysiology is one of the reasons we're going into genomics of schizophrenia is the difficulty, the challenges of understanding the pathophysiology. But there's no agreed histological or molecular pathology. There's no evidence that it's degenerative. There's no discrete lesions and no gliosis. There is some evidence of reduced neuronal size, and reduced synaptic and dendritic markers, but this is a controversial field. Brain imaging studies show, and there are some very nice large studies being done now by the Enigma Consortium, amongst others, and they indicate reduced brain size, widespread reductions in cortical thickness and surface area, and in the size of subcortical structures. There is no particular region of the brain affected. This shows the cortical thickness in reduction in schizophrenia, for example, and you can see that the great majority of the cortex is implicated in this. So, and most of these changes appear to be present at the onset, again supporting, as with the absence of gliosis, a neurodevelopmental type of pathology. There's good evidence that psychosis reflects increased dopamine neurotransmission, and the drugs that we use to treat psychosis block dopamine neurotransmission. And there's evidence over and above what I've already mentioned that this is a neurodevelopmental disorder comes from the fact that people with schizophrenia often have childhood antecedents. Many of the risk factors operate early in life. And the idea here is that there is an interaction between an early, what used to be called a lesion, but early impairment of brain development and subsequent brain development. So as the brain develops around the lesion, if you like, deficits may emerge later as particular functions are called into play and found to be impaired. So moving to the genetics, the genetic, there's been a lot of genetic epidemiology of schizophrenia, a huge amount. I don't know what schizophrenia attracts geneticists like flies, and it attracted the pre-molecular geneticists as much as it has the molecular geneticists. There's been loads of family twin and adoption studies, which mean that we're confident that this is a disorder with high heritability. But that inherited genetic factors are not the whole story. Identical twins have a concordance rate of around 50%, and the risk in first-degree relatives is around 10%. So what about the findings from molecular genetics? And I'm going to talk about copy number variants. I'm going to talk about SNPs and GWAS very briefly, and I'm going to talk about rare variants. So the first sort of set of findings that after years of trying to get somewhere with schizophrenia was the finding, in fact, that schizophrenia is associated with the velocardiofacial deletion, the 22q11 deletion. And that was followed when whole genome methods for assessing copy number variants were developed by findings implicating a range of about 8 or 9 different recurrent copy number variants that are associated with schizophrenia. And about 2.5% of cases carry one of these copy number variants. And these are really fairly robust risk factors for schizophrenia. Well, very robust. GWAS has, well, the Psychiatric Genomics Consortium of Schizophrenia has been one of the big successes, I think, of the sort of collective genomics effort. The paper PGC3, which has been led by, particularly by my colleagues Mick O'Donovan and James Walters in Cardiff, is a study of 667K cases and has found 270 independent genome-wide significant associations. So these are, as you'll appreciate, these are implicating common alleles that have a very small effect on risk. There's evidence from work we did a few years ago that the signal is concentrated in genes that are constrained, loss of function intolerant. There are clearly many more risk alleles to be found, and this accounts, this common variant landscape accounts for about a third of the genetic risk for schizophrenia. So this really is the this brings in rare coding variants to the copy number variants and the SNPs that I've already mentioned. And I should say it includes unpublished data from the Schema Consortium, which is a whole exome study of 24K cases and 100K controls. You can find it on bioarchives, and it together with the PGC3 paper that I showed the data on the last slide, it's wending its way through the review process. But this shows the genetic architecture of schizophrenia, bringing in we've got Schema found 10 genome-wide significant rare variant associations, and they're shown in red. These are the CNVs that I've mentioned before, and here are the common variants. And what you can see is that this shows the imprint of natural selection. Large effect risk alleles are clearly under negative selection, and a nice paper that Elliot Rees, one of our students, did in Cardiff on CNV, showing that these things arise by new mutation, and then they're swept out of the population over a kind of a small single finger number of generations, and replenished by new mutations. There's evidence that some common risk alleles are mildly deleterious. Others are probably neutral, and the idea here is that mutations occur, but a lot longer ago, and then are maintained against negative selection by drift. And this idea is nicely described in this paper by Matthew Keller. In fact, you might ask, is there balancing selection involved in schizophrenia? And we certainly ask that question. It's of course it's possible that there are common alleles that are subject to balancing selection. But by and large, the polygenic score for schizophrenia only is only associated with a very small increase in fecundity in females, and not with males. And this actually increase is insufficient to explain the maintenance of schizophrenia in the population. So at least if you're trying to explain how it's maintained in the population in the face of low fecundity, then balancing selection isn't enough to do that. And it's probably a combination of mutation and drift and mutation selection. Okay, so we've got rare variants and common variants operating together. We've implicated rare and common variants. Do they operate together, or are there some rare variant types of schizophrenia and some common variant types of schizophrenia? Well, it won't, I think, surprise you to know, it didn't surprise us to discover that they operate together. So this is a study of schizophrenia risk CNVs, where we've looked at the polygenic score. And you can see here that CNV carriers who are cases and see it compared with CNV carriers who don't have schizophrenia. And these are their polygenic risk scores for schizophrenia at various p-value thresholds. And you can see that they're elevated. And when you compare CNV cases with non-CNV cases, there's no significant difference. So suggesting that we're dealing here with a polygenic disorder. And of course, there's, as they say, there's nothing new under the sun. And the polygenic theory of schizophrenia was proposed by Irv Gottesman and James Shields back in 1967. And as it turns out that they were correct, as Irv Gottesman used to always tell me. So what about the environment? Well, schizophrenia also seems to be poly-environmental. Risk factors include a number of pre- and perinatal risk factors: stress, famine, infections. You can read these. And interestingly, these factors are also all associated with a range of other neurodevelopmental disorders, things like autism, intellectual disability. There's various biological impacts on children: head injury, epilepsy, which may be an impact or maybe a marker for something else, autoimmune disorders, severe infections. And then there's a range of psychosocial factors, socioeconomic factors, childhood adversity, immigration, and so on. And also season of birth and living in a city are also risk factors. So being born in the spring is a risk factor. And various drugs can induce a psychotic illness that resembles schizophrenia: dopamine agonists, NMDA antagonists, and cannabis seems to be a risk factor. So the odds ratios of these factors are about 1.5 to 3. And of course, one needs to remember the limitations of observational epidemiology, confining, pleiotropy, reverse causation. But you're getting a picture from this of a number of factors that affect the developing brain, and potentially psychosocial factors as well that may be impacting on an already compromised brain. But there's always the question of reverse causation or genetic confounding, and so this is a field that's rapidly evolving. So let's ask, what are the implications of genomic findings for the diagnosis of psychiatric disorders and future research aimed at better strata? Remind ourselves that we have fuzzy borders, especially with bipolar, and that this is probably a neurodevelopmental disorder. So, in schizophrenia, in psychiatry, we still classify them by phenotype, like Carl Linnaeus did. Carl Linnaeus was a man who's not short of self-confidence, and he made this remark: God created Linnaeus, classified her. We don't have Linnaeus, or maybe not even God in psychiatry, but we do have the DSM-5 and the ICD-10 that have codified and made highly reliable the application of syndromic, largely descriptive diagnostic criteria that are highly reliable, but at the expense of the questionable validity. And in some people's minds, often people outside psychiatry, they've become reified. People believe in these concepts as distinct diseases more than is justified by the data. So what do we find when we look at the relationship with other disorders? We find quite high genetic correlations with common variant genetic correlations with other disorders, particularly with bipolar disorder. And one of the really surprising things that we also found, it was surprising to me and to other psychiatrists, I think, was that the CNVs that are associated with schizophrenia are also associated with autism and intellectual disability, because we were taught that schizophrenia was a separate nosological group, really, to childhood disorders, despite being a neurodevelopmental, or potentially a neurodevelopmental disorder. But it's clear that these CNVs, in fact, every schizophrenia CNV is also an autism or intellectual disability CNV. If we look at the relationship with bipolar disorder, well, we proposed with my ex-colleague Nick Craddock and I, that we should really start to view schizophrenia and bipolar disorder as lying on a dimension rather than being discrete disorders. And we've been able to test this with molecular genetic results. Here's a study where we took the polygenic risk score for schizophrenia. We tested a new sample mixture of schizophrenia, bipolar disorder, and schizoaffective disorder. And we looked to see whether the polygenic score for schizophrenia predicted diagnosis. And you can see the deciles here for PRS for schizophrenia going from low to high. Orange is schizophrenia, blue in and is by the two different types of bipolar disorder, and this is schizoaffective disorder. And really what you can see is that as you go from left to right, from low to high polygenic risk for schizophrenia, the proportion of cases with a diagnosis of schizophrenia increases and the proportion with a diagnosis of bipolar decreases. And this clearly supports a dimensional view of these conditions, and you can start to see the potential applicability of these in first episode psychosis cases, perhaps when combined with other variables. The relationship with neurodevelopmental disorders is also something that's interested us, and we've proposed this model whereby we can view these disorders as lying on a continuum, going from intellectual disability, autism, ADHD, schizophrenia, schizoaffective disorder, and bipolar disorder. And this is really a continuum that reflects the severity of impairment to the developing brain that may be genetic or environmental. And there are a number of reasons for doing this, a number of clinical similarities between these disorders. They're all associated with impairments of cognition, impaired social cognition. They're associated with developmental delay in neurological soft signs, early motor abnormalities, and as we've seen with various environmental risk factors that impact the developing brain. And they often, quite frequently, occur together. They're comorbid. And this sometimes gets lost in a diagnostic and clinical practice where clinicians tend to adopt a kind of hierarchical approach to diagnosis. And our prediction was that CNVs and damaging point mutations would be more, there'd be more of a burden of those towards this end of the spectrum. And this has actually been brought out, and it's just been described in a review we wrote recently in World Psychiatry. And what we see is that large CNVs are enriched according to this gradient, loss of function mutations in constrained genes, similarly so. And we also see that genes with rare coding variations in schizophrenia are shared with other neurodevelopmental disorders. So as studies of intellectual disability, the DD study have come out. We've been looking at the overlap, and Elliot Rees, who's going to be talking later about his work in this area, and he's shown that in trios in a paper that came out last year, there's an enrichment of loss of function genes in loss of function, in neurodevelopmental disorder genes, de novo mutations. So these rare CNVs and damaging mutations in constrained genes are associated with a wide range of neurodevelopmental outcomes. And the question is, what determines the outcome? We've some evidence that common alleles modify the outcomes, but it seems likely that there are other rare variants, the environment, and possibly the burden of damaging de novo events. What biological insights do we get from the genetic findings? Remember, dopamine's involved in psychosis. It's a complex multi-domain syndrome affecting many brain regions. And in fact, the genetics seems to confirm this, that it's not unexpected. There's a hit, GWAS hit in the dopamine receptor. Remember, this is the target for the antipsychotic drugs. When we look at the expression of the expression of genes that are implicated by genetic studies, we see that common and rare variant signal is predominantly brain and actually neuronal rather than glial, and that when you look at relative expression in either brain regions or in cell types, you see that this is from 15 different human cell types, and you can see there's enrichment in both excitatory and inhibitory neurons of all sorts of different types. And when you look at bulk expression in different brain regions, you find that there's enrichment in pretty much all brain regions. There is some biological convergence, and this is to the synapse. and I don't have time to go into this in great detail. Here's a review that we wrote a few years ago, showing that the genetic signal, particularly from rare variants, but also from common, is this is an excitatory glutamatergic synapse, and we have convergence onto the postsynaptic density. This is a sort of huge protein machine that is responsible for instantiating synaptic plasticity, and there's a enrichment of rare and common variants in the postsynaptic density and other synaptic components, and this has largely been confirmed by PGC3. By the large GWAS studies. So, you know, it's clearly that the synapse is not the only site of action here, and there are other genes implicated in other processes, but it is starting to point to fundamental biological processes that are related to neuronal function in schizophrenia. So I'm starting to wrap up towards the end, and it's been a whistle-stop tour, but here are the three questions I said I'd try and answer about the nature of schizophrenia. How does it arrive? Well, it's a polygenic and polyenvironmental disorder that affects either damaging mutations or environmental exposures that impact the developing brain. Growing a brain is a pretty complex thing to do, and there are lots of things that can go wrong. But we don't know the major factors underlying heterogeneity in symptoms, course, and outcome. When? Well, there's good evidence that this is a neurodevelopmental disorder, and it's part of a spectrum of neurodevelopmental disorders with things like autism and ADHD and intellectual disability. But when the precise timing of critical events remains uncertain, as does what determines the nature and severity of the neurodevelopmental outcome. You can have two people who carry a damaging mutation, and one may be quite severely intellectually disabled, and another might develop schizophrenia later in life, but have largely preserved intellectual function. Where in the brain does this occur? Well, and I think, and this is perhaps controversially, everywhere. This is a disorder of fundamentally of the way that neurons work. And this, I think, has big implications for downstream studies aiming to connect genomics with brain function. How do we understand what are going to, I think, be many routes from genes to different aspects of the phenotype? But it does, this convergence onto fundamental neuronal processes offers the opportunity for I think, thinking about how to develop new drug targets, although there's clearly more work that needs to be done. Finally, and this is largely from an article that myself and Mick O'Donovan wrote in Biological Psychiatry this year, in a special issue on psychiatric disease. Ours was only a short opinion piece. So how can genomics impact on clinical practice? Well, these are the promises we make. We make them in all our grants, and we've all made them. You know, genomics offers an unbiased route to pathogenesis and thereby to better treatments. It offers the opportunity for prediction and stratification and ultimately precision medicine. How will we get there? Well, we need to do bigger studies and bigger and more. Bigger GWAS, whole exomes, moving on, whole genome sequencing, and we need to identify more risk alleles. And if we do that, then the truth will emerge. And this is really, I think, the kind of dominant genomics paradigm. And I think it has a kind of logic that is hard to, and a momentum that is hard to stop. And once you've found these risk alleles, there are various functional genomic approaches that can be applied, gene set analyses. You can seek convergence onto biological pathways and generate functional hypotheses. And there are some amazing methods being developed, and they're all based on this input, human genetic studies. And I think there are three important concerns to consider. First of all, you know, there's a lack of ancestral diversity, and this will limit applicability and maintain health inequalities across the world. I'm not going to say much more about this because it's been well addressed by, it's being addressed. But importantly, there are two other main concerns. Genomic studies have tended to use prevalent samples, as I've said, from clinical populations, and they oversample severe and chronic cases. And so these GWAS, if you like, a GWAS signal might be, will generate nice biomarkers for the sort of studies I touched on. But they're going to contain signal not just for liability, but for chronicity, including treatment response and non-compliance, disease progression, factors influencing illness behavior, and various confounders that will influence access to care. And the controls are often super healthy. So GWAS signals are not the distilled essence of disease susceptibility. They contain a number of impurities. And it's also, the ascertainment may well affect the number of, the proportion of cases that have rare mutations as well. So we've got this problem with ascertainment. We've also got a problem with phenotyping. The phenotypes that we use in genomic studies are shallower, and they're getting shallower as people move towards population studies. They're also usually cross-sectional. We know in depression that shallow phenotypes are less heritable and less precise than the narrow ones. It seems likely to me that we're going to be missing the heterogeneity of what is a complex multi-domain syndrome if we're not, if we're just making, essentially saying, does this person satisfy DSM criteria for schizophrenia or not? We're also going to miss heterogeneity of course and outcome, which might reveal, in fact, genes for outcome and by processes that underpin outcome might well be the most tractable mechanisms for new treatments. And so my final slide is some recommendations. I'm sure that the more and bigger a school of thought is going to persist, and that we will have more and bigger studies. But I think we need to pause to think about maybe some different types of designs. I think we need to be studying more incident cases, ideally with follow-ups. We have first episode clinics and at-risk mental state clinics that could be used for this. And this will help us with prediction and help us understand the genetics of outcome. We need to have deeper phenotyping, and including in some studies of imaging and biomarkers, to address heterogeneity and disease mechanisms. And we need to do some of these deep phenotype studies at scale using common measures. We need to study the genetics of course and outcome, as well as drug response. And this means we need better measures of course and outcome, and to think hard about what it is that we're measuring in these multi-domain syndromes. There's going to be more population studies and electronic health records, and these will be useful, I think, for understanding, identifying confounders, and will give us access to longitudinal data. But we need to validate the phenotypes against clinical gold standards. And finally, schizophrenia, in fact, other neurodevelopmental disorders are typically underestimated in population cohorts. And I think we can't avoid the fact that we may need to develop specific cohorts for these hard to reach conditions. So thank you very much for your attention.